Hello, my name is Laura and I'm going to introduce you to this project called Dysregulated Alternative Splicing and RNA-binding proteins as potential biomarkers and therapeutic targets for glioblastoma therapy. Glioblastoma is an aggressive type of brain tumor with a poor prognosis. Alternative splicing of prime RNA has been widely reported to be associated with the progression of malignant tumors, including glioblastoma. RNA-binding proteins bind to RNA to control splice site choice and define which exons are included in the resulting major RNA transcript. As alternative splicing has been reported to be associated with the progression of glioblastoma, the main goal of this project was to identify dysregulated alternative splicing events and RNA-binding proteins that may become biomarkers and therapeutic targets in glioblastoma. To achieve this objective, the following pipeline was followed. The first step was to obtain the samples. Specifically, 47 glioblastoma and 36 astrocytoma samples were obtained from the Chinese Glioma Genome Atlas. Once the quality of the samples was checked, transcript quantification and alignment were performed. With this information, we proceed to identify splicing events using different methods. So, the first step was to represent the events identified with each method. This first graph shows the results using event pointer ST. The number of significant events is the sum of the red and blue points, and this corresponds to the last row of the table on the right. It should be noted that a red point indicates that the exon is being excluded in glioblastoma, while the blue ones indicate that the exon is being included. Following the same criteria, in this second graph we see the results when using event pointer BIM. It can be seen that the number of significant events is higher. Finally, with RMAT, it can be seen that the number of identified events is very high and that the number of significant events is similar to that obtained with event pointer BIM. Taking as reference the significant events identified with each method, we match them to analyze the common events resulted in the Venn diagram shown on the right side. Next, by extracting the genes related to the dysregulated events, the enriched biological processes were analyzed, resulting in the two figures. On the left side, the biological processes are shown as a function of the number of genes related to each one. On the right side, the information of the previous graph is extended by including the relation of the genes with the biological processes. Finally, using both event pointer ST and BIM, we obtained that the RBPs represented in the figure were dysregulated, and this indicated possible targets for glioblastoma. So, to conclude, glioblastoma is a highly aggressive type of brain tumor that has a very poor prognosis and sometimes cannot be treated. Alternative splicing of prime RNA has been widely reported to be associated with the progression of malignant tumors. When performing an analysis, usually a comparison of tumor samples versus normal samples is made, as the aim is to study and investigate the differences between healthy and diseased patients. However, one of the main limitations with brain tumors is difficulty to obtain normal brain tissue. This is due to the fact that unless it is needed for some particular reason, brain biopsies are generally not carried out in patients who do not require it. Hence, instead of making a glioblastoma normal comparison, a glioblastoma astrocytoma comparison was made, because although astrocytoma is a brain tumor, it is less aggressive and its prognosis is usually better than for glioblastomas. However, these have resulted in a limitation, as the results have been based on tumor samples and it is not known whether the results will be the same with normal samples. Regarding RBPs, neurodegenerative disorders are the principal clinical manifestation of RBP defects, because of the high prevalence of alternative splicing in the brain, a major function fulfilled by RBPs. Dysregulation of cell RBP family and PTBP1 has been shown to be related with glioblastomas and are very interesting potential targets and biomarkers. Finally, regarding the enrichment analysis, as previously said, glioblastomas are the most difficult tumors to treat due to their high invasive capacity and frequent resistance to apoptosis. With this in mind, it makes sense that one of the most enriched functions is the autophagocyte membrane docking. Even if there are not a large number of genes involved in this function, since one of the main features that make glioblastoma so aggressive is resistance to apoptosis, it makes sense that one of the most enriched functions is just this. Furthermore, looking at the graph in which biological processes are related to the genes, it can be seen that the BMP1 gene appears directly related to this function. This was one of the most significant genes, and its overexpression has been shown to be related with a worse prognosis in glioblastoma. Furthermore, it should be mentioned that many of the genes identified in the different analyses as dysregulated appear as possible interesting biomarkers for glioblastomas. Well, that is the end of this work. There is still a lot of research ahead, but hopefully we can find a way to attack glioblastomas. Thank you very much.